For Review Meeting 4, we're excited to share with you our final project advancements. Our final experimental setup is shown above on the picture of the real-life setup as well as the system diagram on the right. The algae tank is the left bucket, equipped with a fixed light and an OD sensor. The right bucket is the mussel tank, containing a temperature sensor, cooling loop, air pump, and a funnel to hold the mussels. Water circulates from the algae tank to the mussel tank via a pump and returns through a passive siphon. The entire system is thermally and light insulated to minimize external interference and energy consumption. To regulate the tube flow rates accurately, we developed a mathematical model centered on the pump and siphon systems. Flow rates were measured experimentally and fitted to enable reliable predictions. Additional model functions sourced from research papers can also be observed. Using the previously defined functions, we built a model to iteratively simulate cell exchange, growth, and consumption. To account for different conditions, we implemented two versions, one representing ideal behavior and another based on actual OD measurements used in our experiments. Here, we see the outputs of our theoretical model. In the red box, the plot shows how adjusting the pump's PWM duty cycle allows us to maintain a stable algae concentration in the mussel bucket. The blue box highlights the fluid dynamics of the system. Our model accounts for a delay in siphoning as it takes time for the water level to build up enough to match the pump's flow rate. The resulting head development is shown in the graph below. After a safety assessment, we decided to limit the PWM duty cycle of the pump to 50%. This limits the developed head to roughly 3 centimeters. In this manner, we are able to avoid bucket overflowing near our exposed electronics. Afterwards, tuning the model with actual OD measurements, these are the obtained results from a practical model and the overall experiment. To regulate the mussel bucket temperature at 17 degrees, we used the PID controller. The initial 75-minute test on the left showed excessive cooling and oscillations, indicating underdamped tuning. In response, we adjusted the PID parameters, applied a 10-minute on and 2-minute off cooling cycle, and set a 15% minimum pump power to prevent tube freezing. The updated setup on the right achieved stable temperature control with minimal fluctuation. As mentioned in the previous review meeting, all PID regulation was carried out through a web interface made with Adafruit IO. Raw data is available online and offline. With internet access, data is exported and visualized through Adafruit IO feeds, enabling live monitoring of OD temperature and pump power. Without internet access, the same parameters are recorded locally as CSV files. After resolving initial epoxy curing issues, we successfully calibrated the OD sensor through real-life experiments. For each concentration, we recorded 10 OD measurements and used their averages as calibration points. The low standard deviations indicate the sensor provided highly precise readings. By diluting a known algae culture, we prepared samples at defined concentrations and recorded the sensor's response. The resulting data showed a consistent inverse relationship, allowing us to fit a rational function for accurate OD to concentration conversion. We explored reducing the number of pumps by integrating a rotary valve to switch flow direction as shown on the far right. However, the design couldn't be implemented successfully due to 3D printing limitations, specifically difficulty sealing the valve. Support structures interfered with the O-ring grooves, making it challenging to achieve a leak-proof seal. Our idea of waterproofing the OD sensor using epoxy turned out to be successful, especially after the experimental validation that the epoxy was not toxic to the mussels or algae after being soaked in water for long enough time. We were able to achieve a smooth and optically clear finish. A photo of the OD holder is shown on the right. We then investigated the effect of adding PLA on the filtration rate of the mussels. We first measured the filtration rates of 3 mussels under optimal conditions. Then, to test the effect of PLA, we soaked 2 3D printed PLA objects in water for 3 days, diluted the water, and repeated the experiment. Filtration rates dropped noticeably, and one mussel even showed a negative rate, suggesting impaired activity. However, the culture counter had issues during the second test, which may have affected the accuracy of our cell readings. We then looked into the effect of certain materials on the growth rate of algae. After collecting multiple sets of sample data, we compared growth rates of our algae exposed to materials to a control group. We then analyzed whether there was a significant difference between the growth of our control group and our experimental groups. The result showed no significant difference, so we can conclude that all the materials used were safe.